and her focus was on paleontology of primates, so not humans in particular. Afterwards, she went to the University of Pretoria to do a postdoc, and there she got interested in the human fossil record and brain evolution. And she started to study the endocasts, the fossil brains. And she continued with that at the University of Witwatersrand. And afterwards, she's just begun working in Cambridge, where she's a lecturer now. So thank you so much for joining us. I'm looking forward to your talk.
Well, thank you very much, Alexandra, for the introduction. And good morning, everyone. So before I go ahead with the presentation, I just wanted to say thank you to Alexandra for inviting me to contribute that seminar. And so today I'm really pleased to have the opportunity to speak a bit about my research on brain, and more particularly on early hominin brain evolution. So the first thing is, why should we pay attention or be interested in early hominin brain evolution in specifically? So I would say it's because this is related to, in fact, human origins, to the big question of where do we come from. And more specifically, so the fact that we are interested in early hominin, so that is really tightly related to the human origin. So I will explain to you a bit the context. As you probably all know, today we are still not sure about who was the direct ancestor of the genus Homo. So when we look at the hominin family tree here, so what I mean by hominin, it's all the species or the specimens that are more related to us than to any other living species. All right, so here's the hominin family tree. And so you can see here in blue, this is the genus Homo, and we are here, Homo sapiens. And well, you can also see that there's no direct branching from the genus Homo to another, older hominin groups. And the only thing that we know is that the oldest human remains correspond to that mandible found in Eastern Africa, in Mediterranean, and dated to 2.8 million years. So apart from that, so we don't really know what happened during that time period. The only thing we presume, or the main hypothesis, is that potentially the ancestor of the genus Homo would be among the genus Australopithecus. And so you can see this, all these yellow groups, they correspond to some Australopithecus species. So it means that Australopithecus then can be interesting because of this.
Sorry to interrupt. I think the slides are not going through.
You can't see the second slide now.
I think we're only seeing your intro slide.
Oh. So you can't see the where do we come from, who are our ancestors?
Well, only in the side, because it's showing like your whole PowerPoint.
Okay, let me see. I mean, so now if I change the slide, you can't see.
No.
Okay. That's weird.
If you have two screens, it's probably sharing the wrong screen.
Oh, do you have two screens?
Yeah, but the other one is off. Yeah. Okay, let me see. Sorry about that.
No worries. It's tricky. I always forget how to do these things.
I think I would share, because I tried to share only my PowerPoint. Now I'm going to try to share my screen. So maybe that would make the difference. Okay, so if I do that.
But I think it's showing the wrong screen because now it's, oh, okay. So now it's showing again, you're there. That's it. That's the right one.
Then change the
yes. Yeah, this is the right one. Yeah.
Okay, great.
Sorry.
Okay, so I would just, well, this one was not really important. So I was speaking about that one. Okay. Okay, I will start again very briefly. Yeah, so I was speaking about the hominin. Yeah, you couldn't see the hominin family tree, so it's that one. So you can see here in blue the genus Homo, Homo sapiens here, and then we have in yellow the genus Australopithecus. And so I was saying that potentially, so we have the direct ancestor within that yellow group, and so that's why Australopithecus is of particular interest, because it's potentially our direct ancestor. It's also particularly interesting because these hominins, they were living during the what we call the Plio-Pleistocene transition about 2.8 million years ago. And we know that during that time, there were very important changes in the environment and the climate. And so by studying the biology of these hominins, we can also understand how they were adapting to that or coping with these changes. So, and the other groups that is the other group that is of particular interest here is the Australopithecus, the Paranthropus genus. Sorry, so here in reddish, we'll say. And so these hominins, they were living at the same time and in the same environment as the earliest humans. And so here it's very nice for us to try to analyze the different adaptation between humans and the Paranthropus genus, because they were living in the same environment. If the environment changed, then we can try to understand the different adaptations and also the strategies and their evolutionary trajectory. And by doing some comparison, we can also understand better the adaptations that were specific to the genus Homo. So that is why what we call the early hominins, so we say the earliest humans and the Paranthropus and Australopithecus, well, these groups, they can really help us to understand where we come from. And then the next thing is that by studying the brain of early hominins, we can also address the big question of how we became humans. Because if we want to understand, so what were the adaptations that were specific to the genus Homo, then we need to study the interactions between the hominins and their environment. And we can do that by studying there are some interfaces, so biological interfaces between hominins and environment. And here are a few examples of these interfaces. So the first one could be the diet, so the teeth, because of course by consuming some food, so the hominins interacted with the environment because they had to find some food resources. But we can also mention the manipulation, so just by using their hands, they were of course manipulating objects in the environment. But also the development, how they were growing in the environment, and finally the locomotion, so how they were moving from one habitat to another. And so what is also even more interesting here is that all of these interfaces, they are also related to some specific behaviors. So when we studied diet, then we can speak about the dietary behavior. So meaning like if these hominins, they were eating only fruits or maybe meat and they were hunting or scavenging or this kind of behaviors. For manipulation, we can also speak about technical behavior, such as the using or making tools. And for the development, we can also speak about social interactions. So the interactions between the parents and the offsprings, for example. And finally, of course, the locomotion can tell us something about the mobility of these hominids. And in the end, if we think about all of these interfaces and behaviors, they are all related to one specific organ, which is the brain. Because when we speak about the diet, of course, well, through the diet, the hominin could consume food and then provide energy to the brain. And so we can speak about brain metabolism. About the manipulation and technical behavior, so these abilities are also related to some cognitive capacities. The development also, it's very important, and that is also very closely related to the brain, because we know that in extant humans, so our brain takes more time to grow and develop after birth than the other primates. And this prolonged maturation is really important, because this is during that time that we learn how to interact with others, but also to speak or to then manipulate objects. And so finally, the locomotion and mobility can, of course, tell us something about the interactions between the hominin and the environment, and the locomotor behavior is also related to some brain areas. So all of this, that is the main reason why I'm interested in the brain evolution. It's because by speaking about the brain, we can also tell something about the evolution of some specific behaviors of our ancestors. So I was particularly interested in trying to address three main questions. The first one was when and how did the human brain emerge? So when can we see the first the emergence of the very human brain specificities, but also about how did the hominin brain metabolism evolve, and finally about what were the functional implications of such changes. So this presentation will try to give you some examples on these three main questions and categories. But before we really speak about brain hominin evolution, how do we then collect information about brain evolution and also early hominids? So the first thing is, so as I was explaining, if we want to understand the emergence of the genus Homo and the human-like brain, we need to study also Australopithecus and Paranthropus as comparative group. So we need some fossils representing these three different groups. The other thing is that to identify the main specificities of the human brain, we also we need to compare the human brain to the brain of the chimpanzees and bonobos, because these are our closest living relatives. And we know that if we compare the two brains and then we find some features that are specific to the human brain and are not found in the brain of the chimpanzees, then we can say that these features, they are specific to the genus Homo. And on the contrary, if we find some features that are also found in the extant human brain and extant chimpanzee brain, we can suggest that these features, they are inherited from the same common ancestor that we share with the extant chimpanzees and bonobos. So through that comparison, we can kind of assess the polarity of some characters, and we know that some features are really specific to the genus Homo, and we want to focus on them to try to understand how they emerged. So how do we then get some evidence about the fossil records, about these three main genera? So this critical period of the Pleistocene, so about 2.8 million years ago, it's very well documented in Africa. And more particularly in Eastern Africa and Southern Africa, we can find many fossils that represent the earliest humans, but also the genus Paranthropus and Australopithecus. And more particularly, so you will see here that I will speak a bit more about the fossils from Southern Africa, because in South Africa, there is a very well-known region that is called the Cradle of Humankind. And this area is very rich in fossil hominins, and especially early hominins. And there is one specific site, that is the Sterkfontein site that you can see here. This site is has a very high density of fossil hominins. And so more particularly, if you look at the different layers here of that site, so it was a cave. So you have to imagine, so this is part of a paleo cave. And in that sequence, we have very old deposits with hominins from about 3.67 million years old. And we have some younger deposits with hominins about 2.82 million years old. And so this sequence is very rich. We can, for example, in this stratigraphic unit, the member 4, there were more than 800 fossil hominids found, which is a lot. And at the same time, we also have a nice sequence, so we can have very old fossils, fossils that are a bit more recent, so we can really reconstruct the evolutionary trends in some specific features. And the last thing is that at Sterkfontein, the preservation is really good. So you can see here that's the skeleton of Littlefoot. And so Littlefoot is the best preserved Australopithecus skeleton. So it's the best that we have in Africa. So that is why this region is so interesting if we are interested in knowing more about human evolution. So now the thing is, but how do we collect evidence about brain evolution, knowing that the brain tissues cannot preserve? So the only thing we can do is to rely on the study of what we call the endocast. So the endocast is the replica of the inner surface of the braincase. So here you can see three different figures. So that's the brain, that's the endocast, and that's the two together superimposed, and they come from the same individual. So what you can see is that, of course, it's not the replica of the endocast is not the perfect replica of the brain. But at least what we can see is that we have a good idea of the shape of the brain, but also the size, and also the organization, because you can see some imprints of the different cell sites that we can see in the brain. So at least the endocast, it's at least it's something. Unfortunately, it's the only direct evidence that we have, but we can study them and try to reconstruct then hominid brain evolution. So if you're lucky, you can find natural endocasts in the fossil record, such as that one. This is the tongue child, and this one that is from Sterfontein, so they're both from South Africa. And what happened is that during the fossilization process, the sediments were filled in the brain cavity. Well, now the brain is gone, but we can still have the natural endocast, so the kind of natural molding of the inner surface of the braincase. But it's not always the case. So it's very, well, it's not really common to find this natural endocast. So what we can do, and what I do more particularly, is to create some virtual endocasts. So how do we do that? So how do we then use a cranium to get a virtual endocast? So the first thing to do is to use some imaging techniques. So of course we can use CTs, like the medical CTs, but the resolution is not great if we look at very tiny details of the brain, or well, the endocast. So the other option is to use what we call micro CT scanners. So we have an example here. And then with this equipment, we have a resolution about 100 microns for cranium. And the other option, if you want to have a very, very good resolution, you can use a synchrotron. So there is one in the UK, so the diamond light source. And with that technology, then you will have a resolution of about few microns. But well, most of the time, the micro CTs are perfect for endocast. So by scanning then the cranium, what you get in the end is some image stack, and with the images you can see the inner structures of the cranium. So from that image stack, then you can recreate the endocast. And here's an example. So this is an automatic method. So what we do is to use a virtual seed. So we put the seed inside the brain cavity. The seed grows, and then we'll match the inner surface of the braincase. And this is how we can have then a virtual endocast nearly automatically. So now that we have the endocast, so how do we then describe them and quantify their changes, or how do we compare them? So of course, just like for the brain, the endocast is the shape is quite complex. And so the big problem was, but how do we compare them if we really want to get as much information as possible? So what we have been doing is to use surface-based comparison technique. So basically, it's just a comparison between two endocasts, and then we want to see how they differ from each other. So here's an example, just to explain to you briefly how it works. So because here what I want to see is the differences between the fossil specimens and the extant specimens, so humans and chimpanzees, but also between the chimpanzees and humans to detect, so remember the features that are specific to humans. So what we do is that we have the sample, so we see here sample A and B, and we use a template, and the template is kind of deformed to each specimen in the sample. And by doing this, so we have a pretty good information about the variation within the sample or between two different groups. And we can even compute then a taxon shape that is a kind of a consensus of the shape of a specific group. So with this then technique and this information, what we do in the end is to compute some color maps that will tell us exactly where are the main differences in the endocast, or even the similarities between two specimens or two groups, or whatever you want. And the other things that we can also compute multivariate analysis. So that's very quite useful if we want to see, for example, if our fossil specimen is closer to the extant human or to the chimpanzees or bonobos. So that was about the shape. Now, what about the what we call the organization? More particularly, how can we identify the cell type? So here the big problem is that we have to rely on many descriptions, so qualitative then description of the cell site on the endocast. And that was a big problem in paleoneurology, because if you ask two different researchers to describe the same endocast, you will probably come up with two different interpretations and two different descriptions, because it's quite subjective. So depending on your experience with brain or endocast, then you won't describe the same endocast the same way. So to then try to overcome this problem, what we try to develop is then an automatic method to detect the cell site and the imprints in the endocast. So here it's an example of how it works with a human endocast. So the computer, we use an algorithm that is based on the changes on the topography. So in the mesh, you have like some crest and also some ravines, and then the computer will interpret them as the presence of a cell site, for example, of a sulcus, sorry. And so this is the results of the automatic detection. So this is what a computer then can compute. And then in the last step, so we have, at least the scientists, we need at some point to clean manually the results, because you can see here that the detection is so good that you have also the detection of some, for example, vascular imprints. And maybe you are not interested in them, so you have to manually remove them so that you can keep only the cell site, for example. So now what do we know? When we apply all of these methods and study all of these nice specimens, what can we say about early hominin brain evolution? So before I present to you some results about the fossils, there are two main questions that need to be addressed that deal with the endocast. So the first question is, how can we be sure that the endocast is a reliable proxy to reconstruct the brain? Because, of course, there are some differences between the brain and the endocast. So how can we then quantify these differences? So that question is a really tricky question that people try to address for many decades. And the only thing that we could do, before, it was to compare, for example, the brain and the braincase of the same individual, but by doing some post-mortem observations. And so that's, of course, a bit problematic. So what we try to do with that paper, that experiment, is to compare the brain and the endocast of the same individuals, but by using CTs, so for the endocast, and MRIs for the brain. And then by collecting this data, so then we could compare the two. So we could compare the shape, but also the organization of the brain and the endocast. So it took many years because, of course, so we had to collect MRIs and CTs of patients that they do not have any pathologies affecting the brain. That was quite difficult because, of course, it was an a posteriori study. So we just went to the hospitals and asked that, if we could get access to CTs and MRIs of non-pathological patients. And so finally, we could only collect 5 specimens, five individuals, so, which is not a lot, but it's still quite something. So we could compare the brain and the endocast for the morphology first, and we could see that, the correspondence between the two, it's quite good. So the shape of the endocast reflects then the shape of the brain. And you can see here, so that's the differences in terms of distances between the brain and the endocast, so it's quite good, except perhaps on the top of the brain and the endocast. But otherwise, it's quite okay. And then for the organization, so for the imprints and the cell site, we have quite this kind of the same results. So here we computed we wanted to check that if we transfer the cell site of the endocast on the brain, so if we have a correspondence between the cell site and the imprints. And it worked quite well. So just as for the morphology, then we can say that if you identify then the lateral fissure on the endocast, so it's supposed to be at the same position as in the brain. So then we could say, yes, the brain, the endocast is a reliable proxy for studying brain evolution. So that was the first question. The other one was about variation, because of course, if I study 2 different specimens, fossil specimens, and I find some differences in the position of the lunate sulcus, for example, I don't really know if this difference is just consistent with intra-specific variation, for example, or if it means something that potentially we have interspecific differences. So to then address this kind of question, we decided also to study the variation of the imprints of the endocast in extant humans. so that we could build up an atlas, just like for the brain, but here with the endocast. And so we could study the variation, and then the next step that we're still working on is to produce some probability map that will tell us exactly where we are supposed to find a specific sulcus in the endocast. So now what about the fossil record? So as I was saying in the introduction, I'm going to focus on 3 main genera, so Homo, Australopithecus, and Paranthropus. So let's start first with the Australopithecus specimens. So the first thing I wanted to see is if there are any substantial differences in the brain shape between Australopithecus, humans, and chimpanzees and bonobos. So I used the methods that I was explaining, comparing the different surfaces on the endocast, and I focused my study on 2 different Australopithecus specimens from Sterkfontein, so from South Africa, that are relatively well preserved. So you can see here that cranium is quite complete, and this one is a natural endocast. So I could use these 2 and then compare them with the other groups. And what I could find is that the shape of the Australopithecus brain is kind of elongated, so a bit like in extant chimpanzees, and it's well different from the rounded shape of the brain of the extant humans. So that was the main result. However, if we look at the maps here, we can see, well, even if the global shape is similar to the extant chimpanzees, we can still see some differences, and more particularly in the prefrontal cortex. And that was interesting because it means that even if the overall shape is more chimpanzee-like, there are still some differences, and these differences then they affect or they involve some very interesting areas in the brain, and notably the prefrontal cortex. So at least the conclusion was that the overall shape was still kind of primitive, but there was maybe some premises of reorganization in the brain of Australopithecus. Now, what about the imprints and the organization of the brain? So in that study, I wanted to look at Australopithecus specimens, but from different time periods. So that one is the oldest one, and then one about 2 to 2.6, and then one, the most recent one, from 2 million years. And so I wanted to compare them with the extant human and chimpanzees, and also compare each others, just to look at the variation within Australopithecus. So the first thing that we could notice is that at the back of the brain, we can find the lunate sulcus. And so we know that in extant chimpanzees, this lunate sulcus is positioned very more anteriorly than in the extant humans. And this is because potentially the visual cortex is larger in extant chimpanzees than in extant humans. And when we look at the fossil hominins, what we can see is that in that one, the oldest one, so it seems to be very chimpanzee-like, the position, even if we are not very sure if it's really that one. But in the more recent one, potentially the position is more human-like. And so that's kind of, it's quite interesting, because then we can wonder about what happened in between the 2. What happened for that change to emerge then. Then in the frontal lobe, we could also see interesting features in the Broca's cap, or Broca's region. And more particularly, so we know that in extant chimpanzees, there is one specific sulcus, which is the frontoorbital sulcus, while in extant humans, so we have these 2 vertical sulci that define the Broca's cap. And when we look at the fossil, so we systematically see the same chimpanzee-like pattern. So even if the very old one and the most recent one. So at least for the frontal lobe, in terms of organization, we still have something very chimpanzee-like, so very primitive, potentially. Now, what about the brain metabolism? So as I was saying at the beginning, I was also interested in seeing any changes in terms of metabolism. And so that, to reconstruct brain metabolism in fossils, it's not really easy. The only thing we can do is to combine 2 different measurements. One is the size of the internal carotid canal, and the other one is the size of the transverse foramina in the cervical vertebrae. which correspond to the vertebral arteries. So in fact, so we have to, by combining these two measurements, so the size of these two different foramina, then we can estimate blood flow to the brain and also the brain glucose utilization. And so I could do that for some Australopithecus specimens, of course, with little foot, because it preserved the brain, well, the basic cranium, plus also some of the cervical vertebra. I could have a very good idea and good estimation of the blood flow. However, for the other Australopithecus specimens, because they are less preserved, so I could measure the carotid canal, but not the cervical vertebrae. So I use the one of Littlefoot as a proxy, we'll say. So by doing this, by playing with all of these estimations, what I could conclude is that brain glucose utilization in Australopithecus is very consistent with or falls within the range of extant chimpanzees, and it's well below the range that we can see for extant humans. So it means that at that time, the brain metabolism was still also very chimpanzee-like. And that could be interpreted, well, that could be kind of expected, because we know that the brain size in Australopithecus is very small, and it's the size of the chimpanzee brain. So that makes sense, at least. We are not surprised by that result. Now, what about Paranthropus? So for Paranthropus, we don't know much about the brain, because we don't have any, well, we don't have a lot of well-preserved crania or natural endocast. So here I could use only two specimens, one from South Africa here, that is a natural endocast, and another one from Eastern Africa, also weight 5. So well, they come from two different regions in Africa and two different species also. So I wanted to compare them with extant human, extant chimpanzees, like I did with Australopithecus, and also between them, just to see if we can see any regional differences. So the first thing is that the shape, surprisingly, even if Paranthropus is geologically younger than Australopithecus, the brain is here again very chimpanzee-like. So we can see this elongated shape. And the other thing is that if we look at some specific areas, well, the color maps, we can see that it's not, even if it's close to the chimpanzee shape, well, we can still see the differences. And these differences are mainly what could be visible in the parietal cortex. So that was interesting. And it seems that this area is very specific to Paranthropus. And when we compare the two specimens, we can also see that the shape is quite similar between the Paranthropus from Eastern Africa and Paranthropus from Southern Africa, even if they are from two different species. So finally, what about Homo? So for early Homo, what I wanted to see if the frontal lobe was changing within the genus Homo, because the main hypothesis was in the past that there was no big changes in the frontal lobe within the genus Homo, because, well, presumably when the genus Homo emerged, then it already had a very human-like frontal lobe. However, the previous studies were focusing on some 2D sections, so I really wanted to see the changes in 3D. So I could study the frontal lobes of the endocast of 3 different specimens. So here again, I wanted to see, to have specimens from different datings, just to look at what happened and if we could see any changes. So I could then compare them to extant humans using the same technique. And what I could see is that, in fact, we can see some changes in the frontal lobes of, within the genus Homo. And, even if the most recent one, so that one, is very close to what we can see in extant humans, so in the the older ones, it seems to be quite different. So there were indeed changes. And because it's about the frontal lobes, it's very important and very interesting in terms of functions, because we know that there are deep implications for language, memory, or other things. So if we then conclude or try to summarize all of these changes, what we can say is that in Australopithecus, we potentially can see some changes in the occipital part, so in with the lunar sulcus, so in the visual cortex, but the frontal lobe is definitely very primitive. And moreover, what we can say is that when the human brain metabolism emerged after Australopithecus. For Paranthropus, we potentially identify some specificities in the parietal lobe. And for the early humans, what we can say is that we have a late emergence of the modern human-like frontal lobes, because the frontal lobes are still evolving and changing within the genus Homo. But then what all of these changes mean in terms of function and behaviors. So that's why it's so interesting to look at the endocast, because then from the endocast, we can try to interpret then the changes in terms of function. So the fact that we have potentially very early changes in the occipital lobe and the visual cortex in Australopithecus, that's very interesting to, it is to understand maybe how the visual capacities change, potentially in correlation, in correlation with some changes in the environment. And the other thing is that the fact that we have a brain metabolism that is very different from than extant humans, we can potentially suggest that this is related to the diet. And the fact that at that point, Australopithecus were not consuming food, high quality food that would then that would be suitable for having a big brain and then a very important brain metabolism. About Paranthropus, so perhaps these specificities in the parietal lobe could be related to some functional capacities distinct from the extant humans, from the earliest humans that were contemporaneous to Paranthropus. And finally, about early Homo, we can imagine that the changes affecting the frontal lobes could be, why not, correlated to some changes in speech capacity. So this is why, so now just to conclude, the research that I'm doing now is really to try to interpret all of these changes in terms of behavior. And that will also help us to understand how we evolved and our specific adaptations. So now the next step will be really to, now that we know how to extract endocasts and then to get information from the endocasts, so the next step is to try to recreate a fossil brain of hominins. So that's why now I'm working on that step, so try to link the two. So that's, of course, a big project, and we need some different methods of computation, so try to understand the variation in extant humans, potentially to project what we know from the extant humans or chimpanzee brain to the fossil. So that's something we're working on, and also, so we are working on that also with Alexandra and collaborating with some teams in Amsterdam, and just to try to better understand the changes and also to interpret changes we can see in the endocast. And the other things that I really want to relate changes in the brain with changes in social, technical, and dietary behaviors. And so that would involve, some experiments. So we know that some other scientists and teams, they worked on functional MRIs to try to relate to some specific behaviors, such as making tools, to some specific areas in the brain. So by using their results, then we could potentially interpret the changes in the endocast as changes in behaviors, and why not to try to to determine if, for example, Australopithecus was able to make tools or not. And so finally, as I was explaining, I'm also interested in brain metabolism and this potential relation with the diets, changes in the diet. So here the key is to relate cognitive capacities with brain metabolism. And to do that, so I will have then to combine also information about food processing, so it's about dental evidence, but also archaeological evidence with the tools. And so that potentially I could relate to something that is very critical during human evolution, that is the consumption of meat. So the hypothesis is that potentially by eating meat regularly, we could then have access to a high quality food, and that would also help to provide more energy to the brain, and then for the brain to develop and having a big, a larger size and also more complex organization. So thank you very much for your attention. So I would be happy to answer any of your questions. And I would like also to thank all of my colleagues everywhere in the world. And I would like to thank again to Alexandra for inviting me.
Thank you. Thank you so much. So I'll be taking questions. If you want to type them in, I can read them aloud, or feel free to turn on your mic and ask a question. I see someone raising a hand. Oh, Bruce?
Hi there. Thank you for a fantastic talk. And I wanted to ask you a question. Maybe it's one for both you or Alexandra. I have very little knowledge about paleoarchaeology, but one thing which I heard some time ago, which has always really fascinated me, is the report that the human brain has been shrinking more recently, because we have been led to believe, of course, that the Homo brain has been increasing as a function of social interactions and intelligence and so on. But there is this report that the brain shrank around the end of the last ice age, and I wondered if you knew why that was, and is there any evidence of any changes in brain capacity linked to other climate changes events in the record?
Yeah, I think it's really difficult to interpret brain changes in terms of size, because, for example, we know that there were hominin species with a very small brain size, and they were still, while the brain was still very modern, and they were also making tools and doing the same thing as the other hominins. And the other things that we know also, that for a long period of time during human evolution, the brain size increase was correlated to the brain, the body size, in fact. So there is also that aspect. And then, of course, the hypothesis is that at some point, more recently,
Oh, you're frozen.
Looks like the audio got cut off.
Oh no, we just got to the critical bit. Oh. But Alexandra, is it true that there was a significant reduction at the end of the last ice age? That's one thing.
Yeah, so I mean, if I can just chime in, I don't want to interrupt Amelie, but she's gone for the moment. One thing you have to keep in mind is that is based on who gets buried, because if you get buried, either naturally or purposefully, you're more likely to be represented in the fossil record. And those tend to be higher status individuals and males. So that will bring up the average anyway. So that's one thing, I guess, from a more critical perspective, you should consider as well. So while the average is higher, was higher and has dropped, and there are different ideas about, what that means. That data itself, we should be a little bit critical of it for that reason as well. So yeah, so Neanderthals on average had larger brains than the modern person today. So that's and the early modern humans as well. But it doesn't necessarily tell us anything. So it could, but we're not sure yet. So hopefully we'll get more information and be able to understand more about that. I hope Amelie comes back. I'm just going to check if she sent me a message.
Hello again.
Hello.
Hi.
I'm so sorry, my computer just decided to stop.
No problem.
And I
think we got cut off right as you were answering the critical part. I don't know if you were still talking about the implications of the changes in brain size and like the sort of sampling, I think.
Yeah. So yeah, I was thinking about how difficult it is to interpret brain size changes. For the more recent periods, I don't know really if we can relate this to the climate or even also the diet, I guess. So there are many different factors. So yeah, I think that's a good question. Really. So yeah, I think we, for a long period of time during for human evolution, we interpreted the brain size increase as an evidence of increasing in intelligence, these kind of things. I think now we're more and more not reluctant, but we're just really careful with that, saying that potentially, yes, brain organization is also a very important fact, important evidence as well. So yeah, well, I don't know, Alexandra, if you have another opinion.
I already chimed in when you were gone about preservation of what gets preserved in the fossil record for like early humans and Neanderthals. So they're usually like males and things. But so yeah, obviously when we're calculating the averages, I just like when I calculate, I don't necessarily separate that out, and we can't always know either. So these are things to be critical of. Also, we just don't, like you said, we don't really know what that means yet. So we'd like to. We're hoping to get there. I had a question for you too, but first I'll read out the one. I see a couple typed in here. So is there enough fossil evidence with different developmental ages such that you could look at brain development within one extinct species compared to modern humans?
Yeah, that's I think it's a very, very interesting question and yeah, problem. And the problem, as you said, is just the number of fossils that we can have to try to recreate the development and understand the development for the brain. We have some young individuals for example, for Australopithecus. So we know potentially. So there was a new study, I think, last year, concluding that in the Australopithecus afarensis, potentially the development was human-like, but that's based on one specimen. So I'm not sure we can say something just relying on one specimen. And also the other things that we have to be sure about the age and how do we determine the age, then in fossils, etc. So, and even for early Homo and more recent, so taxa, we don't have enough, I think, individuals to really be sure about, okay, we have a development that is definitely human-like. So at the moment, the hypothesis is that potentially it emerged very recently or very early in our evolutionary history. But that's I think it's really a big problem, the fact that we don't have many specimens of younger individuals for that.
Thanks. So a question from Ralph: could the dietary change be associated with cooking, increased nutrient availability resulting from a novel human unique behavior? Would that fit with archaeology?
I think that's a great question, and that's a very important hypothesis. So there is the hypothesis that's the expensive tissue hypothesis, saying that it's only by changing the way, the food we are consuming and how we are consuming food, that could maybe explain that we could evolve such a big brain with very important needs in terms of energy. So potentially, cooking was likely a very important step, because the problem is that with such a big brain, we cannot spend all of our energy trying to digest some different foods, like the early hominins. They were also eating fruits, leaves, and this is very, you need a lot of energy to process this kind of food. So maybe by eating meat and cooking, potentially we spend less energy in trying to digest everything, and we have more energy for the brain. But that's a very hypothetical. So, and it's very difficult to try to falsify that hypothesis in the fossil record, because obviously we don't have we don't have access to soft tissues. So we have to rely on the archaeological record and what we can say about the endocast, fortunately.
Thanks. So I'm going to ask a question. I was really interested in what you said about maybe changes in vision because of the occipital lobe changes early on in Australopithecus. And I was wondering, what sort of, have you thought about environmental factors or anything that, like, why would it change?
Yeah, I think that definitely it's something to do with the environment. Well, that's my hypothesis. I think it's because we know that there were very important changes in the environment during the Paleopleistocene. So especially within the Australopithecus, we know that the landscape changed in Africa. So potentially they had to rely on different food resources and to move in in different environments. So I guess potentially that would have impacted the visual capacities. But it's just a hypothesis. Well, I think that's my hypothesis. But because we have to also rely on the lineage sulcus, and as you know, Alexandra, it's just a nightmare, this sulcus, and there are a lot of huge debates about how to interpret it, how to identify the lineage sulcus, and how we can relate this also to the visual cortex, because potentially that's not a good landmark for saying, okay, that's the visual cortex. So we don't know, but I really believe that the environmental changes really impacted the brain and the different functional areas, and why not the visual capacity.
Another question from Chloe. You mentioned by eating meat regularly, it provides more energy to the brain, developing a larger brain size and more complex organization. Diets that have come into popularization, such as the vegan diet, how do you think it could affect brain size in the long term?
How the diet?
If people become vegan.
Vegan, yeah.
Yeah.
So that's I think. No, now the main problem was to find high quality food. I think now we don't have anymore that problem because we know where to find very high quality food, even if it's not meat. So it won't be a big problem. It's just that that time during human evolution, that was the main source of energy that they could use. But today that we know where to find the energy and what kind of food to eat. So I mean, even if you are vegan, I'm not sure it's a big problem for your brain. And I think it's a very different our diet today. It's very different from diet from a Australopithecus or Paranthropus or Homo.
Thank you. So any more questions? Please feel free to just speak directly too.
Is there evidence for differences in between humans and chimpanzees in visual capabilities? I don't know if it's not really about the extinct hominins, but if there's differences between the visual cortex between the two extant species, do we know, say, whether chimpanzees have some kind of visual ability that goes beyond ours?
I think only about the visual cortex. So we know that the proportionally, so the chimpanzees have a larger visual cortex than the extinct humans. But then the visual capacity. So I don't really know if how we can interpret that, because I guess it's also something to do with the size of the orbits and everything. But if we speak only about the brain, then the visual cortex is supposed to be bigger. But I don't know if Alexandra, you want to add something, because
I think
you are the expert on that.
a bit of my area. So like functionally, visual acuity in chimps and humans is both super high. So they're like the highest of any mammal, both species. It's the same. So actually, we probably have like visual ability, as far as we know, is identical for in terms of ability. There are some sort of peculiar differences in the way that chimps and humans perceive the world, like how they respond to stimuli, like where they think shadows are coming from. So there's something called shape from shading, where you try to determine where the light is coming from by looking at a shaded object, and this differs between humans and chimps. But again, these are not a lot of these sort of cognitive tests have been done on really, really small samples, and so maybe it doesn't represent all of chimps and all of humans. But generally, the idea is that it's actually really, really similar, which is why I thought it was so interesting that things could have changed really early on with Australopithecus, and they have like the really small, if they have, in fact, the lunate sulcus in that position. It's a question that, really interesting to me as well. Any other questions? So thank you so much. It was a super exciting talk. I'm sorry about the technical glitch at the beginning, but they're beautiful slides. So it was really nice. Oh, it looks like someone, well, maybe she was leaving actually. I thought someone popped in. If anybody has any more questions, otherwise I think we're gonna end now. Please speak up. Okay, well, thank you so much. It was really lovely to have you.
Thank you. It was great to virtually meet you.
It's fun. I'm going to turn off the recording.